Let's talk about the nervous system, an AP Biology review with Amelia Rusli. The nervous system is divided into two parts, the central nervous system, or CNS, and the peripheral nervous system, or PNS. You can remember the CNS because it is in the center of the body. You can remember the PNS because it is in the periphery, or everywhere else. The CNS consists of the brain and spinal cord, and the PNS consists of all nerves not in the CNS, so cranial and spinal nerves. Alright, peripheral nervous system. The first part of the peripheral nervous system is the sensory system. This transmits information from sense receptors or nerve endings to the CNS. So, as you can see, we're getting the water and we're transmitting that information to the central nervous system. The motor system is the other part of the peripheral nervous system, and this controls your muscles and glands. The motor system is further divided into somatic and autonomic systems. The somatic system controls voluntary muscles, and the autonomic system controls involuntary muscles. Out of the autonomic system, some nerves are sympathetic and are part of the fight or flight reaction, and others are parasympathetic and calm the body down. These parasympathetic nerves are going to oppose the sympathetic nerves. So here's a little picture of a cat. This fight or flight is sympathetic, and this relaxed is parasympathetic. Alright. The neuron. The basic parts of the neuron are the cell body, nucleus, dendrites, axon, and axon or synaptic terminal. Surrounding the axon is the myelin sheath. The myelin sheath is composed of glial cells, sometimes they're Schwann cells, and these little gaps in between the glial cells are going to be called nodes of Ranvier. These are very important, but we will discuss that later. All types of neurons, they are sensory or afferent neurons, are going to receive stimuli from sense organs and transmit the information from sense receptors to the CNS. Motor or efferent neurons are going to stimulate effectors, which are muscles or glands. They are going to transmit information from the CNS to the muscle or glands. So you can remember these by the acronym SAME. SA for sensory afferent and ME for motor efferent. And interneurons are located in the CNS and receive sensory stimuli and transfer the information directly to the motor neurons or the brain. So, let's remember this sequence. The stimulus is received by the sensory neurons, transferred to the interneurons, and then returned to the motor neurons. Very important. Keep that in your brain. Reflex arc. This is inborn, it is automatic, and it is protective. So an example of the reflex arc is the knee-jerk reflex. When you go to the doctor and they tap a hammer on your knee, your nerves are going to control your response. The hammer tapping is going to be the stimuli, which will be picked up by the sensory neurons and transferred to the motor neurons, which will cause the muscle contraction. So science is everywhere. Resting potential. Now that we understand what is transferring information, let's take a closer look at how the information is transmitted. Membrane potential is the difference in electrical charge between the cytoplasm and the extracellular fluid. When a neuron is at resting potential, it is polarized and has a charge of -70 MV. A stimulus must overcome a threshold and leave the resting state to send a message. Make sure to note that at a resting state, the extracellular fluid is more positive than the intracellular fluid, and we'll see why this is important later. Alright, so the sodium-potassium pump. This is very important for maintaining resting potential. It pumps 3 sodium ions out for every 2 potassium ions in, allowing for a depolarized state to return to a polarized state. Speaking of depolarization, let's talk about hyperpolarization versus depolarization. So what we're comparing here is the direction of movement of positive ions, such as sodium or potassium, as they move across the membrane. In hyperpolarization, positive ions are going to move out, and the difference in charge between the sides of the membrane is going to increase. This is because the cytoplasm is normally more negative than the extracellular fluid. So if even more positive ions flow out, the cytoplasm is going to become even more negative. For example, the cell might go from negative 70 MV, which is resting potential, to negative 75 MV. Alright, so in depolarization, the opposite is going to happen, and positive ions are going to flow into the cell. Depolarization is what leads to the generation of an action potential, which I'll explain later. Alright, so yeah, there's positive ions moving in. Alright, gated channels. Before we get into action potentials, let's take a minute to look at some gated channels. These are going to open and close in response to a stimulus and are going to allow one kind of ion to pass. So unlike the sodium-potassium pump, which transports two kinds of ions, these channels are ion-specific. Alright, so let's look at two of these. The sodium ion gated channel looks like this, and as you can see, there are multiple different states it can be in. Before the stimulus is sensed, the channel will probably be closed like this. But when the channel is activated, it will open. And so the potassium ion gated channel is very similar, but when it's activated, the membrane potential will become hyperpolarized to negative 75 MV. This will make it harder for the neuron to fire and continue the message. Now let's talk about the star of the show, action potentials. These are an impulse that are generated in the axon and consist of a wave of depolarization. Action potentials are going to reverse the polarity of a membrane. What I mean by this is that the charges of the intracellular and extracellular fluid are going to reverse. So, if you look at the diagram, the cytosol starts off negative. But during the rising phase of the action potential, it becomes positive. Then, when the potassium channel reopens and the sodium channel closes, the cytosol returns to a negative charge. In this diagram, the deactivation of the sodium channels is labeled as undershoot, but we're going to refer to it as the refractory period. The refractory period is when the sodium-potassium pump returns the membrane to its original polarized condition. It only lasts a few milliseconds, but during this time, the neuron cannot respond to another stimulus. It is put on hold. Alright, so how does potential travel? Each action potential generates the next action potential. It's kind of like dominoes. If you knock one down, then the rest are going to fall down. As the action potential travels, it does not lose any strength. It remains strong. However, sometimes it can travel faster in the presence of a myelinated axon. So, remember how we talked about the myelin sheath and nodes of Ranvier? Well, an action potential is going to travel by saltatory conduction from node to node. Basically, all the depolarization that I've been talking about is going to occur at the nodes of Ranvier, and the action potential jumps from node to node. Alright. Have an all or none property. What this means is that there is a threshold. If the membrane potential reaches the threshold, then an action potential will be generated. But if it doesn't reach the threshold, no action potential will be generated, and the message will not be sent. This helps the body distinguish between weak and strong stimuli. Alright, now let's talk about the synapse. It's important to note that while most signals in the nervous system are transmitted electrically, the synapse transmits information chemically. So let's walk through the steps of the signal being transmitted across a synapse. Alright, so first, the action potential is going to reach the action terminal, and then the action potential is going to trigger the calcium channels to open, and calcium is going to rush in. Then, the calcium pushes these synaptic vessels full of neurotransmitter to the presynaptic membrane, where they release neurotransmitter. Once the neurotransmitter is released, it travels across the synapse, and when it gets to the postsynaptic membrane, it binds to the neuroreceptors, which are proteins. By binding to these neuroreceptors, they are triggering a signal in the postsynaptic neuron membrane. Alright, so, of neurotransmitters. I'm only showing these so that you can recognize them, but you don't need to stress too much over the different functions and whatnot. So as you can see, we have acetylcholine, serotonin, epinephrine, norepinephrine, and dopamine. Just make sure you know that these are all types of neurotransmitter. However, it is important that you know the difference between excitatory and inhibitory. Excitatory will depolarize the postsynaptic membrane and stimulate the brain. Inhibitory will hyperpolarize the postsynaptic membrane and calm the brain. In other words, excitatory is going to continue the message and inhibitory is going to inhibit the message. The thing I'm going to cover is the human brain. I would highly recommend going into more detail as you study, but for now I'm just going to cover the 3 most basic parts of the brain. First, we have the cerebrum, which controls learning, emotion, memory, and perception. This is the part of the brain that is divided into right and left hemispheres, so the expressions right-brained and left-brained are going to refer to the cerebrum. Each hemisphere controls the opposite side of the body. Below the posterior of the brain, we have the cerebellum. This controls homeostasis, so breathing, swallowing, and digestion. The cerebellum receives sensory information and transfers the information between the PNS and the brain. Last, we have the brainstem, which controls movement and balance. It censors the position of joints and muscles and uses motor commands from the cerebrum to maintain coordination. Whew! Alright then, well that's it for this lesson. I really hope everyone enjoyed it, or at least learned something. I'll let you enjoy these memes for a minute, and that's it. Thank you very much.